Proteins are condensation polymerization from amino acids, so your monomers are amino acids. If I put a generic amino acid, by generic I mean instead of spelling out what this radical is, if it's CH3 it's alanine, if it's this segment it's arginine. I'm going to put R to represent that, COOH. When you put two monomers next to each other and they line up, they are going to go through condensation polymerization because each monomer has two functional groups. I put R prime. R prime could be same as R, or it could be different representation. So this is condensation one more time. Condensation among the hydroxide of the carboxylic group and the hydrogen of the amine. So a molecule of water is going to condense out, you lose H2O, and the remainder becomes dipeptide because you simply have two amino acids. So what you're going to form is a new functional group, C double bond O, N H, C H, R prime, C O O H. Now this functional group that you formed is known as, in organic chemistry we refer to it as amide. In biology or biochemistry you refer to it as peptide. So this is formation of amino acid, condensation polymerization. Your monomers are simply your amino acids. Now proteins have four different structures and we need to know that. The first one, primary structure, is the sequence of amino acid. If I put the four amino acids next to each other, one possible combination is alanine next to arginine, next to asparagine, next to aspartic acid. But you could have many combinations. You could have alanine and simply put arginine in different placement, and this strand is totally different from the one above. So the question is, how many possible proteins can you make when you have four amino acids? And we go by the rule of factorial. 4 factorial, which is mathematically same as 4 times 3 times 2 times 1. And this is 24 possible combinations or proteins. So the sequence or the order that you put amino acids together is the primary. Then you have other structures. Secondary structure comes when there is a alpha helix possibility, or when the strand goes into a shape like this and it becomes beta pleated. In both cases there is a hydrogen bonding between the hydrogen of nitrogen and the oxygen of carboxylic group. So in both cases you have the same hydrogen bonding going. And this is what makes this strand to go into secondary structure, possible alpha helix, possible beta plated, so H bonding. Now this strand or this molecule of protein can further fold into itself and give you a three-dimensional arrangement in space, and it has many possible interactions. You could have hydrogen bonding, you could have simply London dispersion, if your amino acids have sulfur, which is cysteine, you could have disulfide breach, and you could also have a ionic bond between the amine side and the carboxylic side, which are in ionic version of them. So you could also have that. And you have to be able to look at a structure and pinpoint what are those interactions. So hydrogen bonding, London dispersion, disulfide, ionic are possibility for this strand to fold it into itself. Finally, the last structure of proteins is when you have more than one molecule interacting together, and that's when you have two possible strands or many strands that are working together, and that makes it quaternary structure of proteins. So when you have individual strands that are cooperating and working together as a protein. To have a review, so primary structure is when amino acids in a sequence are going to go and form a polymer, condensation. Now this strand could fold into itself to give you alpha helix, or it could fold in this fashion and give you beta pleated, and the hydrogen bondings are going to hold them in place. This strand can further collapse into itself and have hydrogen bonding, disulfide bridge, ionic interactions, London dispersion to give you tertiary. Finally, you could have more than one strand or one molecule of proteins reacting together and acting together, and that's known as quaternary structure of proteins.